with your
due
OGT,
to
whatever.
increase in colloid. So colloid goiter is usually due to lack of iodine in your diet. It is referred to as an endemic goiter, so that's another term meaning you've got this lack of iodine in your diet that's causing the colloid to build up. You can't get enough iodines on the thyroid molecule, the thyroidine molecule just keeps getting recycled. Plus the cell is still being stimulated by TSH because T3s and T4s are not adequate in the circulation. So TRH will be high, TSH will be high, and a thyroid will be stimulated to produce more binding protein, more thymine. Try and get this T3 and T4 back up, but you're lacking the iodide in your diet. You'll have a hypothyroid condition. T3s and T4s are below the normal circulating levels that they should be in. You're going to have a very particular phenotype with this individual. You're going to be obese because thyroid hormone drives your metabolic rate. If you're not stimulating your metabolic rate, then you have excess calories coming in. They're getting stored in the fat, and fat cells enlarge here and become obese. The metabolic rate is low, so all these covalent bonds that are supposed to be broken to make heat for your body, because we're very inefficient at using the energy for work. We use it for body heat. So you have this obese person who should be warm because they have all this insulation, but they're always cold. They have 2, 3, 4 sweaters on. They're always complaining about being cold. You touch their hands are cold all the time. Their feet are cold. Their whole body is cold. So this is not what you would expect with an obese person. You ask them what they eat, they say, I don't eat at all. I eat like a bird. And you're thinking, yeah, sure you do. But then you actually monitor what they're eating. They'll put them in a clinic situation and monitor what they eat. They have no appetite because they're not burning calories. They consume very little, but what they do consume is not being utilized, so it's stored as fat. And they keep gaining weight. They're lethargic, they don't want to move, they may sleep a lot. So all of this is typical of hypothyroid condition. Could be you're lacking iodine in your diet. And so solution is get some of that iodized salt and sprinkle it on your food. Get some more T3s and T4s out there. Get your metabolic rate back up. Start burning some of those excess calories. Shrink the fat cells down, get the energy level back up, throw off some of the sweaters, and get back a normal life. But there are conditions where it's a problem with the cell. And so when I was first learning about colloid goiters, I looked up what causes endemic goiters besides these colloid goiters besides lack of iodine in your diet. And it turns out people who OD on mustard, people who overdose on yellow turnips, and then people working in labs that come in contact with propylthiouracil that will cause your thyroid cells to be unable to convert iodide to iodine, and so they can't, there's no iodine to put on the molecule. I don't know, I've never run into anybody who ODs with mustard on their hot dogs. I mean, you have to eat a lot of mustard to do that. And I don't know yellow turnips from white turnips, so I don't know of anybody who overdoses on those, but obviously somebody must be or it wouldn't have made it into the literature. But if you're working in the lab, oftentimes there's PTU, propylthiouracil, in your lab, and you really want to be careful to keep it away from you. You don't want to ingest it. You don't want to have contact with your bodily fluids because it will create a hypothyroid condition. I want to tell you, I went to a workshop on the mainland, and there was a professor on the mainland who was teaching a lab on taste. And she had this method of determining whether an individual was a super taster or just had normal taste buds. And she was handing out litmus paper that was saturated with trophothioid uracil. She said, put this on your tongue. And I'm thinking, no way, Jose, I'm putting that on my tongue. Because I don't know how much she put on that litmus paper. If there's enough, I could develop a hypothyroid condition. So I started wanting to see what it was all about, so I tore up a tiny little piece of the litmus paper, figuring that's not probably that much. I stuck it where it's supposed to stimulate your bitter receptors on the back of my tongue, and it's like this gag. She goes, "Oh, super taster over there." So it was doing what it was supposed to do, is give you this gag response to this bitter, the chemical bitterness, but be careful. And I also thought that was really not a very bright thing to do with undergraduate students who might get too many of those litmus papers and then tell their friends, oh, put this on your tongue. And just start layering it on kids' tongues, and then you'd be surprised what kids will do. And, oh, let's make our friends gag. So a little bit, let's put more, it's even better. Okay, so PTU definitely will be long-term hypothyroid condition if you have that ingested, so be careful. Hyperthyroid condition. You also get an enlarged thyroid, but this is due to overstimulation of the thyroid and overproduction of T3s and T4. So you have a completely different phenotype. You have somebody who's very skinny, no muscular mass, no fat mass. They're hyperactive, they can't sit still, they're just jittery all the time, they're eating all the time, they're hot, so they'll be walking around with minimal socially acceptable clothing on. And if it's been happening for a long period of time, the eyes tend to protrude forward out of the head. So it's, if I can pronounce it, it's something like exophthalmos, something like that. It means your eyes are bugging out of your head. That's a sign of hyperthyroid condition. Could be that there's a tumor in the thyroid, and so any little TSH coming causes an overproduction of T3 and T4. It also could be that negative feedback is failing. So a tumor in the thyroid, negative feedback is working. You'll have high T3 and T4 because the thyroid gland is producing too much, and low TRH and low TSH because negative feedback is working. When negative feedback fails, that means you do not have a problem with your thyroid gland. You have a problem that is either due to something like Graves' disease, where you have a circulating antibody that's mimicking TSH. TSH is not high, TRH is not high, but the thyroid gland is still overproducing thyroid hormone because this antibody is binding to TSH receptors and keeping the thyroid gland producing T3s and T4s. Could be you don't have Graves' disease. Maybe it is a problem in the hypothalamus. If TRH is high, and that's because of a hypothalamic brain tumor, then TSH will be high and T3 and T4 will be high. Normal feedback is to shut off TRH. Negative feedback is probably turning off TSH, but TRH from the tumor is coming and re-stimulating TSH release. So TSH will be high, TRH will be high, and T3 and T4 will be high. So you know there's a problem in the hypothalamus. It's a brain tumor. Do the blood workup and you see high TSH and high T3 and T4 but low TRH. Then you suspect anterior pituitary tumor. So the TSH cells, they're too numerous. They're producing too much TSH. Even with very little TRH stimulation, they overproduce TSH. The thyroid gland responds to that as it should. It's responding normally to high TSH, producing high T3 and T4. Feedback to the hypothalamus is working. TRH is low. Feedback to the anterior pituitary is not working because you got tumors that are overproducing TSH. So if you remember this when you go into medicine, you will not be the dumb doctor on the medical mysteries who scratch their head. I don't know what the problem is. Well, you check out the blood workup, and it will tell you, based on the concentrations of these different hormones, where the problem is arising. So everything is high. Probably look at the hypothalamus. If T3s and T4s are high, but TRH and TSH is low, look at the thyroid gland. Either you've got a tumor there or you've got antibodies overstimulating the hormone. So you remember your basic 103 physiology. You will not be a dumb doctor when you get out there and into a clinic situation, or a dumb med tech, or a dumb nurse, or any other medical profession. Okay, if you are going to treat hyperthyroid conditions, sometimes surgeons will go in there and remove part of the thyroid so that you have less secreting cells to secrete thyroid hormone. Be very careful when you do that to check out how many parathyroid glands that individual has. Supposed to have 4, but not everybody has 4. If they have 2 and you remove those 2, that person will die. If they have 1 and you remove that 1, that person will die. If they have 4 and you remove 2, they'll survive. But you have to know, because these are migrating, they're tissues that are migrating from your 4th pharyngeal pouch down to the superior lobes of the thyroid, or the 3rd pharyngeal pouch to the inferior lobes. If that migration tract goes astray, then those cells don't reach the thyroid. They tend to get reabsorbed. They just become atretic and disappear. And so you don't have the full complement of parathyroid. If you don't have parathyroid, then you can't elevate your blood calcium levels. When it gets too low, your muscles go into tetany. If your respiratory muscles go into tetany, then you suffocate to death. So you don't want to remove somebody's last parathyroid gland. It's attached posteriorly, has its own capsule, which is embedded in the thyroid capsule, so you actually see space around the parathyroid. Thyroid will have this colloid, this amorphous gelatinous mass with cuboidal cells usually around it, and then the parathyroid will be strikingly different. A lot of densely packed purple staining nuclei. Those cells are the principal cells, and then they notice that as the organism ages, these other cells, these larger cells, start to appear, and those they looked at them histologically and saw a lot of mitochondria there. So they knew that they were using a lot of oxygen. They called them oxyphil cells, cells that are liking oxygen, so oxyphilic cells, but they just call it oxyphil cells. They knew that they appeared as the organism got older, didn't know for a long time what their function was, and it was really quite a mystery and interested a lot of people because it's a super high metabolic rate cell, but what the heck did it do? Turned out that these are older versions of principal cells. As principal cells age, they accumulate more mitochondria, consume more oxygen, and continue to do what parathyroid cells do. The principal cells release parathyroid hormone. So mystery solved, they're just older versions. That's why they appear as the organism gets older, more of these oxyphil cells show up. You see them in the parathyroid because you notice them because there's more space around their nucleus. The nucleus is a little bit larger, a little bit paler staining, and then huge cytoplasm around it. Look at high magnification, you see the increased number of mitochondria there. Fenestrated capillaries here, fenestrated capillaries in the thyroid. Again, you're moving TSH out to stimulate and thyroid hormone into the bloodstream. Here you have calcium being the stimulus for parathyroid hormone release, so low calcium sensed by the principal cells, parathyroid hormone is released. The C cells in the thyroid respond to high calcium levels. This third pharyngeal pouch also, summarize this. This is just a summary slide, so pretty much covered all of that. I won't go over that again. Again, thyroid hormone is there to drive your metabolic rate. So if you're deficient in thyroid hormone, you're going to be obese, you're going to be cold, any extra calories, any calories you do take in will probably be stored as fat, leading to the obesity. So it drives your basal metabolic rate. Also, then anything, any additional movement that you do above the baseline, lying on your back awake after a 12-hour fast, that determines BMR. And you'll have resting and active metabolic rates also that rely on thyroid hormone. So where the gland, what the gland is, where the cells, specific cells are, what hormones come out, what the targets are, and what the effects are. So you review that in table 19.2. So thymus also is from the third pharyngeal pouch. So as the parathyroids are descending, the thymus is also descending from that third pharyngeal pouch. Goes past the thyroid down into the thoracic cavity to form the thymus around the level of the heart. The thymus is releasing the thymosins that promote the education of the T cells. So the lymphocytes now can recognize major histocompatibility complexes that are your self-antigens. They are doing that early on, and as you get towards teenage years, most of these T cells that have passed the test, they're out in other immune tissues in the body, so they can undergo mitosis elsewhere in the body. So they're not, the thymus isn't needed for their proliferation anymore, and it starts to involute, it starts to shrink and become more fibrous and have more adipose in it. Adrenal glands, these are sitting on top of the kidney. They're attached to the kidney, so ad meaning attached to, suprarenal meaning on top of the kidney. They have their own arteries that go to the cortex, to the capsule out here, and then branch and start to filter through the different layers of cells in the cortex, then down into the medulla region. In the capsule, you just have connective tissue with the arteries. In the outermost layer of the adrenal cortex, that's the zona glomerulosa. The cells there are located or they're arranged in clusters, circular clusters. So that circular structure is called a glomerulus. We're going to see glomerular structures in other areas as well, like the kidneys will have a round capsule, and we'll have a ball of capillaries that will form a glomerular structure. Here, the cells are clustered in these glomeruli. The blood vessels that come into the capsule will then branch and go around these cells, and they'll collect the hormones that are being secreted by those cells. In the zona glomerulosa, you have aldosterone being released. Aldosterone, so aldo here, is being released due to angiotensin II arriving. Aldosterone will head back to the kidney that's underneath the adrenal gland. So this has to go through the blood, out to the systemic circulation, heads back to the kidney, and you increase the reabsorption of sodium. So downstream here, in the kidney, you had a decrease in blood pressure, which is due to a decrease in sodium and decrease in water. These two make up your ECF. That's your major component of your extracellular fluid. So if you lose sodium, you lose water, your blood pressure drops. Blood pressure drops, sensed by the kidney. Kidney releases renin, which is an enzyme that cleaves an inactive angiotensinogen to angiotensin 1. That goes to the lungs. You get angiotensin 2. Angiotensin 2 goes to the adrenal cortex, and aldosterone is released. Go back to the kidney, reabsorb that sodium, water follows passively, and you you're correcting your blood pressure. The next layer of the adrenal cortex, the cells are lined up in rows. So we had other things that are lined up in rows. They form muscle fascicles, nerve fascicles. Here we have fascicles of the secretory cells. Blood vessels branch and go between these rows of cells in the zona fasciculata, so fascicles of cells. Zona fasciculata is releasing your glucocorticoids, cortisol and corticosterone. What causes those to release their hormones is ACTH from the anterior pituitary. The third layer in, the cells are kind of going all different directions, forming a network, which is called, if you remember back to your connective tissue, reticular tissue is a network of collagen, fine collagen fibers. This is a network of cells, so it's called the zona reticularis. And zona reticularis will appear when you're approaching puberty. So 7, 8, 9 years of age, this zone starts to appear. It's releasing small amounts of androgens that then in the males will get converted to testosterone, in the female to estrogens. Then the blood vessels, they're kind of going in a network through here. Then they finally get into the adrenal medulla, and now you've got cells that look like cell bodies of neurons, because they are cell bodies of neurons, just no axons. Those will release norepinephrine and epinephrine, mostly epinephrine. That goes into the blood in the adrenal medulla, and then that gets collected into larger veins, and you're out into the systemic circulation. So a summary of what the adrenal gland zones do: so glomerulosa, mineralocorticoids, glucocorticoids, androgens, epinephrine, norepinephrine. Target for aldosterone is the kidneys, glucocorticoid, most cells, and then your androgens for the various sexual characteristics that your testes and your ovaries would also be producing those androgens for. But the adrenal medulla backs up your nervous system, saves you a lot of energy currency for your nervous system, can turn on your nervous system, recruit organ system responses quickly, but then as soon as the epinephrine and the norepinephrine arrive, then your autonomic nervous system, sympathetic side, can dial back the nervous system, and you maintain the function of your nervous system with the hormone arriving. Pancreas, you have most of the pancreas is for next semester for digestive purposes. The dark purple staining makes up cells make up the majority of the pancreas. Those are exocrine pancreas cells. They release into the ducts that then will release into your small intestine, your first part of your small intestine, the duodenum. What we're focusing on now are the endocrine cells. They have lost their duct system, and blood vessels then penetrated the cells, and now the cells are releasing into the blood vessels. Those are the whiter areas that you see in between the pale staining cells. These pale staining cells are forming little islands in this purple mass of exocrine cells. So they're referred to as islets. So little islets, pancreatic islets. Also sometimes referred to as islets of Langerhans, but they're trying to stay away from that. But you'll see that in the textbook. By the way, this used to be called islet of Langerhans, just in case you forgot. You don't know what a pancreatic islet is, it's the islet of Langerhans. The islet contains a lot of different kinds of cells. The main ones that most textbooks focus on, of course, the beta cell, which is the most numerous. It releases insulin. Next most numerous are the alpha cells that release glucagon, and then the F cells and D cells that are less numerous. The beta cells tend to be more in the center of the islet, the alpha cells more in the periphery, and then the others kind of all over the place. But insulin secreting cells tend to be in the center of the islet. The pancreas is developing as an evagination of the duodenum. So it's growing out of the duodenum. Also at that same point is the liver growing out of the duodenum. You'll see the bile duct from the liver and the main pancreatic duct from the pancreas going to release bile and enzymes through this main hepatopancreatic ampulla and sphincter. Some people have an accessory lobe, and so they'll have an accessory pancreatic duct. Many people do not. They just have this one main duct that goes into the duodenum. The islets appear very early, and at 20 weeks, you can actually detect insulin in the circulation of the baby. The organ, again, looks nodular like the thyroid. It's got these bumpy, extra outside surfaces. It's going to be located between the duodenum and the stomach. The stomach will form the first part of the duodenum. The duodenum then wraps behind the stomach, will actually pull the pancreas with it behind the stomach. So stomach, pancreas, and then duodenum. Most of it, as I said, is exocrine, so that will be digestive, and that will be studied next semester. The endocrine part, the alpha cells, you got about 15 to 20% of those. They tend to be more in the periphery of the islet. Majority, 70%, are beta cells, and they tend to be in the center of the islet. Next most numerous is delta cells that release the somatostatin. Somatostatin has various functions. They know it's released to turn off growth hormone, but probably this is not released in high enough concentrations to actually get into the blood and affect growth hormone in the systemic area. But what it does do is it turns off insulin secretion and glucagon secretion and also pancreatic polypeptide secretion. So it's turning off the islet secretion. When it's high enough, then somatostatin will have receptors on its cells for the various secretory products, and when there's enough stimulation of the D cell, then somatostatin is released, and that then turns off the beta cell or the alpha cell or the F cell. F cells, pancreatic polypeptide, those that will be studied next semester in the digestive system. So again, summary of what's going on in the pancreatic islet. And glucagon is mainly released during periods when you haven't eaten for 6 to 12 hours. So 3 hours after meal, glucagon's not high, but you're approaching 6 hours, and then overnight fast to 12 hours, glucagon will start to rise. We'll see more about glucagon in Sherwood's chapter. Glucagon is going to try and elevate blood glucose back up. So it will mobilize fat stores, it will have the liver release glycogen initially, just nuke glucose right away out of the liver, but then it will also make new glucose out of anything it can get its hands on. So you're not using your skeletal muscle, it will break that down for amino acids to make glucose. If you're using your skeletal muscle on a regular basis, that spares it from catabolism, and the body will go after fat. And then if you and then use beta oxidation to make new glucose out of the carbons in the fatty acid chain. And then somatostatin will reach the digestive system and also slow down absorption and slow down secretion in the digestive tract. So it sounds like it might be a bad thing, but you want to control absorption of certain nutrients so you don't overwhelm things like insulin levels. You don't want blood glucose to go up so high that the insulin isn't adequate to handle it. You don't want enzyme secretion very high, because then you have a lot of these metabolically active molecules that may pull water out into the digestive system. You don't want them creating a gradient for fluids to go on through the digestive tract and just cause diarrhea downstream. You want digestion to happen slowly enough that you can handle the absorption. Okay, testes and ovaries, summary here, more about it next semester. So testes The interstitial cells start with androgens converted to testosterone, which drives sperm production. The sustentacular cells will pass testosterone onto the spermatocytes that are developing. They keep the blood from outside the seminiferous tubules separated from the sperm production inside. So you have these nerve cells that will support the sperm production, but also then sense whether it is adequate or not. If the sperm production is adequate in that region, then the sustentacular cells will release a chemical inhibin, which will turn off FSH and LH release from your anterior pituitary. In the ovary, the LH will drive estrogen production. Estrogen is its own, this estradiol is its own inhibin. When the follicle gets big and filled with estradiol, starts to leak out into the blood, and that goes back and turns off FSH and LH secretion. After ovulation, the cells that remain, the follicular cells that remain, and the cells that were making androgens early on, they mix and form a whole new endocrine organ called the corpus luteum, which will produce progesterones that will then go to the uterus and get the uterus ready just in case there's fertilization and you might need to have the uterus support a developing embryo. Okay, so pineal gland, we don't have time to cover it, so we'll cover it after next week's exam.